Director of the Institute on Ecosystems, and this is our first RepCut seminar of the year. So welcome and thank you for coming. Paige is logging on to the WebEx on her laptop, and you can use the chat there if you have any feedback you want to give us about what's happening with the technology. And before I introduce the speakers, I just want to share a few housekeeping items. I want to remind everyone that we have lots of ways for us to keep in touch with you. We have a MSU faculty email list. We have a big email list that goes to the whole state network for the Institute on Ecosystems. And then we've also just started one for graduate students. So if you're not getting her emails and you want to get them, just let Paige or I know. And then I really do want everyone to know that some very exciting news has transpired this summer. The University of Montana has appointed a new co-director of the Institute on Ecosystems, Dr. Frank Rosenwig, and he will be here to meet IOE-related faculty and students on Tuesday, September 14th. And we're going to do a coffee hour in the Trapphagen conference room from 8 to 9:30 a.m. And I will keep reminding you of that, but I'd love to see you there. Finally, before we get started, I want to thank Paige Tolson for all her work organizing the series and for being the varsity tech athlete that she never thought she was going to be. So today is the first in a whole series of talks that help us as scientists and practitioners begin to continue to tackle the dilemmas associated with climate change. So our whole RepCut series this fall is really devoted to that in various dimensions. And I'm really excited to welcome Vern and Ford Smith from Regenerative Land Solutions. And just there's a little bit of feedback, so if you can make sure you're on mute if you're visiting us from cyberspace, that would be great. So Vern and Ford Smith are from Regenerative Land Solutions. Vern is a longtime rancher from the Paradise Valley and has been managing land holistically since the 80s. His deep dive soil health journey began with a Ranching for Profit seminar in 2004. Son Ford grew up on the ranch along the Yellowstone. He studied economics and political science at Williams. He owns a soil health-based landscaping business and is a whitewater kayaking fan. Together, we're going to learn about their passion for healthy soil. And so welcome, Vern and Ford, and thanks for putting up with these new times. We're so glad to have you.
Thank you. Thank you. Yeah, we appreciate the opportunity to talk. My son and I started this business a few years ago. Like you said, that Ford's a kayaker. He likes to kayak in clean waters, and we figure if we can help people manage for biology, perhaps that over time we can diminish our chemical footprint on the land, and clean things up. So our main goal is to try to view growing crops, any kind of agriculture, landscaping, it doesn't matter, through a different lens than what's historically been used. And typically it's been a chemistry first approach, and we're trying to see if we can't shift that paradigm to a biology first approach. Up on the screen, these little guys, they're the workforce underneath the ground. If we don't manage for them, they'll leave. They're free. They work 24/7. So we try to measure them. We try to figure out how we can help them do a better job for us. We're working with a lab out of Corvallis that was started by Dr. Elaine Ingram years ago, and it's called EarthFort. She was one of the first proponents for the soil food web. If you look at the screen there on the left is the first trophic level for plants, how they get their food, and then the second trophic level, that's the area that we're trying to do most of our work in. Just a little background for some of you guys for plants, and this is our understanding. This is a constantly evolving science, but when a plant needs something in a natural state, it'll secrete sugars, it'll attract the different free-floating fungi and the bacteria that they're out there mining the different minerals, and they'll bring them back to the rhizosphere of the plant. At that point there, we have protozoa and nematodes. When they come, they'll prey on those little, basically fertilizer bags. That's how I kind of view the fungi and bacteria. When they prey on them, and then when they secrete them, that's perfect plant available nutrients. Problem is, if you give that plant free food in like a chemical form and give it salt and force its uptake, that plant's going to start disassociating with the microbial community, which in turn, those little guys aren't going to die. They're survivors. So they'll end up eating more of the organic matter. This goes back to the climate change, and that's carbon. And we don't want to see that happen. We want the plant to have a full association with the microbial community, and we feel we can help people get that through some of the work we're doing. I'm sure a lot of you guys have seen this particular slide, but it shows how small percentage that those organisms are, like 0.1%. But a key thing is air and water. We need to have that in there. And when I get a little further along, I'll explain how these the bacteria and fungi can help make that better for the soil. So if you don't have 25% air and 25% water, the soil is not healthy. And that's what we're striving for. Organic matter, it can be anywhere. We just put 5% on there, but that varies. We're constantly trying to get that number higher as we go. Most of these agriculture, lawns, landscaping, whatever it is, anytime we're manipulating the ground and working on it, we're disturbing it. The thing that we disturb the most is the fungi. The fungi is the delicate part of the microbial community. And so we want to learn how to mitigate that and not be so tough on it. And this particular diagram talks about plant succession as a relationship to fungal bacteria ratios. And we'll talk about that more in cropping systems. So in nature, Mother Nature never likes to be she wants to be covered all the time. So what you have a disaster, you have a volcano, you have a mudslide, that ground, the first type of biology that's going to come on the scene is going to be bacteria, followed by the fungi. And so the plant community reflects that ratio there. So if it's a really bacterially dominated system, the early successional plants in nature are going to be like, there's a lot of different ones, but in agriculture, they end up typically tending to be weeds and annual plants. Over time, as the fungi builds up, then you get your perennial grasses. You get more fungi, and that ratio gets higher. You get your shrubs, and then you get your trees. And then in the forest, you could have a ratio that's 100 or 1,000 to 1. Where we want to be, and on this particular slide, it says 1 to 1, but anywhere from 1 to 1 to 1 to 10 is where we want to be. But every time we disturb the soil, whether we do it through tillage, chemicals, salty fertilizers, it's going to set us back. And so we end up going back to a system that's bacterially dominated. And that's not what we want for the plants we're growing. And I might come back to this, but no, I probably won't actually. So positive ripple effects of good soil. When I talked about how the plant gets its nutrients, the plant wants to decide what it needs. It doesn't need a full dose of whatever it needs in the spring, because some of those things are going to get tied up. Some of them aren't going to become available to the plant. So if we can allow the plant to decide what it needs, when it needs, let the soil feed it, and then we can microdose our nutrients later, we can have a more nutrient dense crop. At the same time, if we have healthier soil, we're going to not lose our carbon source. Every molecule of water will attract 16 molecules, or every molecule of carbon, will attract 16 molecules of water. We don't want to lose any of them. We also want the fungi building the macro aggregates to give the space for the air and to give the space for the water. In these dry conditions, if we don't want to lose rain, we don't want it running off, and we don't want it running through the soil, we want the plants to hold it. And the fertility and germination rates, they also increase. There's a lot of studies that indicate that driving force in germination isn't fertility, it's the fungal bacteria ratio. We'll have an example or two of that coming up also, and it's a different way of thinking. It's lost on a lot of people. Disease and insect pressure are trials we're doing in California and Florida And even in Illinois, they're showing that the healthier the plants, the less disease pressure you have and less insect pressure. I know, for example, last year we had a really bad grasshopper year, a lot of places, and we're still having the same deal. I treated my fields last fall pretty heavy, try to get the biology better. And this year, the grasshoppers are leaving me alone. And a lot of that is because the plants are healthier. So if you have healthier plants, if Brix is the way you measure sugar in plants, is BRIX. Well, if you can get your Brix count up, grasshoppers don't like that. They don't like sugar. It poisons them. In fact, I have a lot of grasshoppers hanging dead on tall grass in my field. They just didn't make it. And that, to me, that's a positive deal. It's also a system of diminishing inputs and greater profitability. If you can get your system more robust, you can start backing off on your fertility program. And eventually, if you can get to a protein-based source, a lot of your fertility you put down is not going to be washed away or end up in the water table or the streams. The best medicine we have on the planet is healthy food. Well, let's grow better, healthier food. Clean water, we just talked about that. The soil filters water. The more biology is in there, the cleaner the water will be coming out, and the less chemical we have to put down. So then, therefore, we're not polluting as much, which is it's a win-win for everybody. And weed pressure, which we'll have some examples of, is also a bonus. There'll be a lot less weeds. The fungi we talked about it plays a pretty significant role in soil structure because it's the heavy lifter in the system, which builds soil structure. It builds macro aggregates. So when we looked at the soil food web, there's your protozoa, you have your amoebas and flagellates. Well, the flagellates like bigger areas and the amoebas like smaller. So if you don't have a good fungal bacteria ratio, you're going to be losing some of your key players when it comes to nutrient cycling. Anyway, when I look at soil, we do some testing, we do direct microscopy testing with that lab and Corvallis, and the driving forces that I see is the fungal bacteria ratio and then the active fungi to total fungi ratio. That means how much of that fungi is actually working for us. We had a producer in the Paradise Valley he had this place for 30 plus years. He tried to do things right. He just wasn't getting the production he needed. So we said, let's take a soil test. So I'll walk through this real quick with you guys. I don't have a pointer, I don't think. Yeah, I do. I can't figure that out. So I'll just ignore the top stuff, but when you get down to the total fungal to total bacteria ratio, TF to TB, it's 1.28. We talked about on that one slide. That's pretty balanced. But then when we drop down to the active fungi to total fungi, which is about halfway down the page, it's 0.03. He has basically sleepy soils, not much activity going on. So when you look at his flagellates and amoeba and you take in consideration the population of his fungi and bacteria, those ratios indicate that his nitrogen cycling potential is about 100 to 150. In most healthy fields, we want to see that number up to 300. Well, he's doing half of what he probably should do. So we worked with them. We boosted up a system, and we have two products. We have a compost tea and then a food for the fungi. If you can feed the fungi, 90% of the time, the bacteria will follow and be healthy too. So we bumped him up, and he's doing well now. He's pretty happy. And he didn't have to do it through chemistry. And that's one of our goals, is to get, if he wanted to get an increase in production, yeah, he could have thrown a bunch of chemical on there, but he didn't want to do that. And we're hoping people will look the same way he does at things. This is over by the airport. Good example of the way the fungal bacteria ratio relates to what you're going to see with your crops. He planted this before we got to know her. So the field to the right in the lower right-hand corner had a 0.39 fungal bacteria ratio, very bacterially dominated, be very supportive of weeds, but not supportive of a perennial grass. The top left-hand, it was 1.27. So she seeded that. We had a good rain. She probably had a 60% success rate on the field that had the better fungal bacteria ratio. And then the other field was beautiful Standacotia. None of her grass germinated. It wasn't really what she wanted. If she had tested before she did this, we could have helped mitigate that for her. What's interesting is this is a 20-acre plot divided by a canal. Previous ownership, we don't know what their practices were, but for some reason, one side of the ditch soil was pretty dead, and this is in the fall. I'd be pretty happy with that top left-hand corner, especially since it was under non-irrigated conditions in the first time seeding. Pretty disappointed in the lower right-hand corner, but she's changed that around for herself. This picture looks kind of confusing because it looks like the treated side didn't do as well as the untreated side. But what we've got going on here is another example of how a bacterially dominated system makes annual grasses. So Texas rescue grass down Texas is a southern version of our cheatgrass. It's an annual non-native bromus family. And I don't think anyone wants a full stand of cheatgrass, and she didn't want a full stand of rescue grass either. So we planted some perennial grass seed in there. I'm looking at the picture on the left. We didn't treat that, and the rescue grass did really well. We treated the right side. In that picture, you can't see it, but there's a lot of perennial grass coming up. The rescue grass didn't want to germinate, so we were able to outcompete it with biology by shifting the system further up the ladder of succession. We're seeing some pretty cool stuff in Montana where without using chemical, we can knock back some of the cheatgrass. Cheatgrass is not, it's a huge issue. We haven't done anything with Medusa head, but I think it might too. Might work on that. No one wants soil like this. But we had the feds decided to put a new bridge in by the house. I worked with the guys building the bridge, and then this is substrate from the county road on the truck route down in Tom Miner Basin. It's gravel. It hasn't seen air or daylight for long time. I don't know how long that road's been there, 100 years minimum. So I thought, you know what, we filled a hole. I thought I'd try something. So we have a compost tea with the food extract. So I thought I would use this for an experiment because it's a small area. But you guys know the wind blows down into Paradise Valley. So I had a hydro seed company come, and I said, I want you to just put a thin layer down. So I used a 12 seed cover crop mix with the perennial grasses I wanted. And then I had them mix it in with the hydro seed deal so that you couldn't even see it. It was that thin, but it glued it to the ground so it didn't blow away. And I thought, you know what, I was busy. So I mixed them up enough for their tank, and I was getting ready to leave. And he goes, what about the other 3 loads? Well, I didn't realize it was going to take him 4 loads to cover the area. So I put 4 times the product down on the right of that picture than I did on the left. And the perennial grass is established on basically a gravel parking lot, and they're still there today. So you can, biology doesn't need a plant, but a plant needs biology. We're pretty convinced of that, and this is a pretty good example of that. This is out in my front fields. That hillside for 20 years wouldn't grow grass very well. So I had 2 treatments on the left side in 2 years, and I didn't treat the right side. And this is in spring, right when the grass is starting to grow. So the grass came on stronger early and also thickened up, and it's still that way today. Have another example on this deal too. So I also did the one part is my neighbors, and so I did where I did the treated and the untreated. We took some soil samples. Dr. Eberly came and did that in 2018, and he put it in a deep freeze, and eventually came around to testing it. So on the side that I didn't treat the bacteria, he didn't do a DNA sequencing on the fungi, but he did a series on the bacteria. But what was interesting was, and I knew I had an acidity problem, but the soil on the untreated side had bacteria indicative of poor, unhealthy, acidic soils, and this soil on the other side that I treated had different bacteria, complete different DNA of healthier soil. So that system shifted in 2 years of working on it, and I no longer take care of the one field that was the control, but I did go ahead and I treated a lower part of it. So it's been 4 years since I've done anything to that field. It hasn't had water. Right now, everything that I didn't treat is kochia and pigweed, and where I did treat, the grass is still hanging on 4 years later. I'm not saying it's going to hang on forever, but it's kind of interesting. I should have taken a picture of that, but I didn't. Jed Eberly wrote this slide up for us here, but I just explained that. Living Sky Greens is owned by a guy named Frank. We work with out in towards Three Forks. It used to be the Wheat Montana farm. He bought it. He's really in to trying to figure out how biology can help him. And he did 14 trial plots last year, different biologicals. He worked with us. Our particular plot didn't get the best deal because he got everything out, and then they had that big snow last year, and then so then he had to wait for the fields to dry. So we got put out on some dry ground, and he factored that in. But even in spite of that, even because it didn't rain after that, last year was a disaster for these guys. So his production is not what he'd want it to be, but the product we use did give him more production. This year's not as good as last year. This year's worse. It's drier. But he did say in a conversation recently in the email that things aren't good, but where he's using the biology, his inputs were 30% less. So he's still winning not using conventional chemistry. Plants are plants. I have the neighbor up the road really loves her flowers. She was struggling to get flowers to grow. So I said, if I'll help you, I'll give you some stuff, just spray it on and let's get some life there. But she had a promise not to treat one plant. So the picture on the top left-hand corner is 2 plants she sprayed, and the one in the lower right-hand corner is one that she left. I mean, it exploded. My father-in-law had the same thing in Key West. I was down there, and he loves his plants, and they didn't look good. And so I sent him some stuff. He didn't read the directions. He thought it was 1 pound per gallon when it was actually a tablespoon. And that was my fault. I should have made it clear. But his place turned into a jungle. So their biology, managing it pays, definitely pays. So I'm going to turn this over to Lila right now. And this is some of our trials she's going to explain that we're doing down in the Paradise Valley.
Hi, everyone. My name is Lila Rickenbaugh. I have been helping Ford and Vern over the summer through a fellowship from Dartmouth, and will now be working for them full time starting now, I guess, September. So I'm just going to be building on the foundation Vern just laid and talking a little bit more about some of the trials that we've been working on, both in Paradise Valley with our neighbors, and also some of the stuff that we've been working on in California with an almond grower. So the first of those is the church pivot trial, which we've been working on at AMB West. The idea behind this study was to test a biological approach against a traditional approach side by side on a blank slate. So that means a new seeding. And this is giving us a more accurate picture than looking at a biological approach on top of years of a traditional approach. So this is a 40-acre pivot on an alfalfa grass field. We split the field in half, and on one side used a biological approach, which was treated with Earthfort's Provide and Revive. Provide is a biological inoculant that Vern was talking about above. It's made by the company named Earthfort out of Oregon and made from a wild source rather than produced in a lab. It's a diverse and abundant microbial spectrum, and Revive is the food source for these microorganisms that also has humics to break down salts. We also used fish hydrolysate on this side of the field, which is an organic-based nitrogen source, and G22, which is just a less salty and more biologically friendly fertilizer. On the other half of the pivot, we had a traditional chemistry approach, which was Rocky Mountain Fertilizer's status quo fertility program. We applied both of those treatments in the spring of 2020. And just to note here, the biological program was $7 cheaper upfront. So we took a soil biology test in the spring before we had treated either side of the field just to get a foundation for what we were working with. And pre-application, we found that the soil had really low fungal activity and was too bacterial for an alfalfa grass field. Like Vern was speaking about above, these conditions favor annual weeds. So as you can see there, the total fungi to total bacteria ratio was 0.46, and it really should be between 1 and 10. One of the big goals of this trial was to look at weeds on either side of the pivot. And so in the first summer of growth, we established this sampling layout where we tossed 6 hoops on each side with 50 paces between each random hoop toss. And then we counted every weed in the circle. And in just the first season of growth, the visual difference was pretty striking. On the left side, you see the biological approach side of the pivot, which is largely grass and alfalfa. And on the right side is the traditional chemistry approach, which is there's more pronounced yellow mustard. And just the visual difference was pretty striking. But when we actually looked at the numbers from counting the hoops, we found that the total number of weeds per acre on the traditional side was more than 4 times higher than on the biological side, which again is just a pretty remarkable difference. We also wanted to look at feed quality in this trial. And so at the end of that first season of growth, we took a feed analysis from both sides. And what's really important here is that we didn't lose any quality in feed, but actually gained a little bit of quality in the biologically treated side. So you can see we've highlighted the crude protein percentage here and also the relative feed value. And in both of those metrics, it's slightly higher on the treated side. It's also really important to look at plant tissue when you're looking at the difference between these approaches, because as nutrients cycle in the soil, what's really important is that those nutrients make it to the plant. And what we observed here after the first season of growth was that the biological approach had healthier plants. We saw higher nitrogen and potassium levels in the brome tissue on the biological side. And so it seems that the higher microbial activity is making those nutrients more available to the plant. Also, in this first season of growth, we found that the biological approach was allowing a more robust cover of the cash meadow brome and the alfalfa, which means that there was higher germination and success rate on the grass on the biological side, which is really important because we are looking to have the least amount of bare ground on a field. The less bare ground you can have especially on a new field, the more production you'll get both in the short term and the long term. In the short term, because obviously you have more grass and alfalfa, and in the long term, because you're less vulnerable to weed invasion. We also added a fall application of biologicals to put this field to bed, that in the fall of 2020. Biological programs work really well in the fall as the microbial community can build upon itself until the ground freezes and then wake up early in the spring. So now this past summer in 2021, we looked a little bit more at our production and did a hoop sampling estimate with a local NRCS agent. It should be noted in this slide that these are the averages from only 2 hoop tosses. So we were really just learning how to use the hoop toss to estimate production and also seeing what we could see on this trial. So these results can vary. But in the sample that we took, we saw almost 50% more cash meadow brome on the biological side than the traditional chemistry side. And the alfalfa was almost equal on both sides. So again, that's consistent with our observations from 2020 that the biology supported germination and success rate of the brome and alfalfa over annual weeds and left less bare ground. And lastly, these are just some images from this past 2021 summer. In the top left, you see the traditional chemistry side of the pivot, which has more weeds and bare ground and less alfalfa and grass. And in the bottom right, you see the biological side of the pivot, which is more grass and alfalfa. This trial is really promising because it's suggesting that the biological program we're working with can maintain production with lower inputs per acre and without using any chemicals. We've also been working with our neighbors in Paradise Valley to trial this biological approach on their hayfields. The first of those trials that we're going to share is on an old 25-acre grass alfalfa field. The producer hasn't fertilized this field in a couple of years, and basically we did a strip trial. So we left one strip in the middle untreated and then treated both sides of that strip with the same program. That program was 1 gallon per acre of that fish hydrolysate, the organic nitrogen source, 1 gallon per acre of Provide, the biological inoculant, and 1 pound per acre of Revive, which is the food source. We took a soil sample in late June before the first cutting. And again, this is one snapshot in time of a really complex system. But we did find some promising results just in that first, first year of that microbial community being sort of jump-started. And we found a total fungi to total bacteria ratio of 0.87 on the untreated strip and the total fungi to total bacteria ratio of 3.9 on the treated section. Alpha grass should have, again, a total fungi to total bacteria ratio between 1 and 10. And so that treated section is hitting the mark and the untreated section is not. We also looked at a production estimate in this trial. We tossed just one hoop for either side into representative stands and found that the treated side had 30% more grass and 61% more alfalfa than the control. And our visual observation also confirmed that. And then just an anecdotal piece from another one of our neighbors, a producer we've been working with in Paradise Valley for about 4 years. We were on the phone with him a few weeks ago, and he said that since he started using this biological program, he's seen a 25% increase in production each year and expects that increase again this year, which is remarkable because of the drought this year. So he will have effectively doubled his hay production in the 4 years since he changed his fertility program. And lastly, I'm just going to run through some trials that we've been doing with an almond grower in California who's on his way to transitioning to regenerative. We set up this trial in 2019 to understand whether a biological approach can improve soil health, improve water holding capacity, withstand disease pressure without reducing yields. So we established 4 treatment blocks and split them into 3 treatments and 1 control. You can see the specific components of each treatment here, but basically the treatment number 1 was fully organic using Provide and Revive, that biological inoculant that we've been speaking of. The treatment number 2 is fully organic, but using a worm tea extract. Treatment number 3 is basically a conventional program with Provide and Revive on top. And the control is just a completely conventional program at 100% fertilizer rates. In the first year, in 2019, we found no differences in yield between treatments, which is great. And on top of that, we found successes in the total fungi to bacteria ratio. In treatment number 1, that metric improved by 78%. And in treatment 3, that metric improved by 48%. Treatment 1, just to remind you, is the one that's fully organic using Provide and Revive. And treatment 3 is Provide and Revive on top of a conventional program. And for treatments 2 and 4, the fungi to bacteria ratio worsened by 58% and 25% respectively. So both of those treatment programs were ones that did not have that biological inoculant. This ratio is really important to keep track of when growing almonds because as a deciduous tree, almonds require more fungi than bacteria, and the ideal ratio is between 5 and 10. So treatments 1 and 3 are closer to that metric than 2 and 4, or the control. Our visual assessment of the soil health also showed significant improvement of tilth structure, porosity, drainage, water holding capacity, and organic matter building in the treatment blocks that were using that biological inoculant. And here we just have some images of trees from the block in the treatment 1 block, which is that organic program using Provide and Revive. We continued this trial into 2020. However, the grower wanted to change some of the trial locations and treatments based on the successes and failures of the previous season. So this next slide here is just kind of a summary of those changes. In the first treatment, we added fish hydrolysate and combined the locations from treatment 1 and 2 from 2019. Treatment B is in a new location, and we added fish hydrolysate and foliar applications of herbicides and pesticides at 70% rates. Treatment C is entirely new. It's using a worm tea extract and has foliar, pesticide, and herbicide applications at 70% rates and is in a new location. And treatment D is also completely new. There's that biological inoculant and food source, fish hydrolysate, and the standard fertilizer program at 50% rates, and that's at a new site. So you can basically think of treatment A as regenerative, treatment B as an organic program using Provide and Revive, but with foliar applications, treatment C as organic using worm tea extract, but with foliar applications, and then treatment D as standard fertilizer plus Provide and Revive. And the results from this second year of growth were a little bit more significant and interesting than the 2019 results. In all 4 blocks, again, yields were maintained from historical averages, which is really important in all of these side-by-side trials. All 4 blocks had noticeably increased nutrient efficiency, which is really important given that they were treatments B and C, at least, were at 70% of normal NPK rates, and for treatment C, 50% of normal rates. So it just means that with decreased inputs, those nutrients are cycling more efficiently. Treatments B, C, and D were significantly cheaper than the old conventional program, and treatment 1 was just more expensive because of the high amount of compost applied. And lastly, all the Provide and Revive treatments had improved water holding capacity. When you harvest almonds, you shake the tree, and for that to happen, you need the tree to be dry. And normally that takes about 7 days, about a week, for the tree to hit that drought stress enough that you can shake the tree for harvest. And in all of the treatment blocks where Provide and Revive was applied, it actually took 21 days before they were able to hit that drought stress and shake the tree. So I will hand it over to Ford to talk about Tomcat.
Hello, my name's Ford Smith. I'm going to build on what the basics that Vern talked about, and then these trials that we've worked on together. And then these next 2 examples are projects we did with academic partners, so they're a little more rigorous. First one was with Tomcat Ranch. This is on rangeland. Tomcat Ranch, they're on coastal California. It's a grass-fed beef operation, about 1,300 acres. And they have the Tomcat Ranch Educational Foundation put up some money for this, and we worked with Dr. Chelsea Carey of Point Blue. We set up a replicated study in 2018. There were 10 replicates. So we picked 10 fields over the course of this giant ranch, and we wanted to see if we could increase feed and forage quality. Obviously, they want to have happier, bigger cows, fatter cows. And then we also were curious about these more academic questions of, are these things actually changing the microbial community? So we tested biomass. We had exclosures set up on the corners of our plots, and we moved them yearly after we sampled, and everything kept getting grazed like normal. And then we collected forage quality as well. And then we did the soil biology direct microscopy tests, like the ones Vern and Lila had talked about earlier. But then we added in DNA sequencing. So we did that for both bacteria and fungi. And we sampled spring and fall for 4 years. So I'll end up with a lot of data. And this was all, this was all unirrigated land. So we found for the 3 years, we were able to get good biomass data. We found that increased feed on all of these fields where we were treating them. That last year was definitely the driest. I mean, California's had a bunch of dry years in a row. So we did see increased biomass. For forage quality, here's an example from 2019. We saw increased quality in forage. Noticeably, they were only marginally significant. So, we had a lot of replicates, but we are seeing it trending up. What was interesting is on this particular one, we didn't see as much fungal and bacterial metrics changing as we had on some of those other simpler trials we had earlier. And then with the DNA sequencing, we did not necessarily see the changes that we had maybe hoped we might have seen, but we have a lot of thoughts on this. I mean, this is healthy rangeland that probably already had healthy bacteria and fungi populations. It's a little different than when you're working on an almond orchard where it's heavily disturbed, or corn soy where they're heavily disturbed in the Midwest. So some takeaways from this: we did see the forage production go up and the quality improved. The DNA sequencing shows there wasn't so much. We have so much still to learn about DNA sequencing and how these biological products work. We definitely need more research in this area. This paper is currently being peer reviewed, should be released here soon, we're hoping. But what's really interesting about this one is we've paused it now. We're not going to sample again this fall. We're going to take a year off and come back in 2020. A lot of these studies are 3-4 year studies, but nobody ever goes back to look at them. This one, they've got a plan to come back in 2020 and do more sampling to see what the lasting results are. And then, well-managed unirrigated rangeland might not be our lowest hanging fruit. This was the first study that we ever really got set up. But we came back from that and decided we wanted to see what it would do here on irrigated land. We might get more bang for our buck, and also on land that's already getting some fertilizer. So we worked with Dr. Jed Eberly in MSU, and then this is AMB West again in Paradise Valley. We set up a replicated study. We had 2 treatments. We wanted to test the difference between, we think of that as just 2 different groups of biologicals, Provide or the worm tea. They're very diverse. They come from wild sources. Then there's a whole other huge industry of biologicals that are made in a lab, where they pick specific strains of bacteria out that they think might work. So we tested those 2. Spectrum is made by Tanio. They make them in a lab, ones that they think will work. So we set this up. We also wanted to do compost, but after the first compost application, we realized how hard it is to source compost down in Paradise Valley, and we nixed it. It didn't seem like it was a practical thing for producers to be using down there. So we had 2 sites for this. We had an irrigated alfalfa field, and then we also had an irrigated pasture. Both of these, this was back in 2018, so we put it just on top of the conventional fertility program. Ideally, we would have maybe made some more changes like we did in the other ones, but this is just biology on top of their status quo. Did an initial fall application, 2018. We did some 2019 and post cuttings, and then again in spring 2020. So we took all these, when we mix the compost, you can see like the compost plus spectrum or a compost plus provide. In the subsequent years, they just turned into more provide or spectrum replicates. So we had 6 replicates of each. We took metrics, we did the sequencing, which we're still waiting to really go through all that information. It's a lot. We took soil chemistry, and then we also did the direct microscopy testing, and then forage as well in biomass. So here's some interesting things that pulled out. Jed helped us make these charts, but the organic matter went up. We started this presentation is all about climate change and soil carbon, and managing for biology we think does store more carbon. So you see soil organic matter went up, and then we also had more nutrients freed up as well. You can see the phosphates there. Provides in the middle, spectrum's on the right. And then we also analyzed, from our clippings, we saw that the dry weight went up, so there's more feed on those, the diverse provide ones, and then as well as both of the biologicals increased the relative feed value. So that's what producers want to hear. I mean, they're interested in those deeper questions, and we are too, about what exactly is going on in the microbial community. But a lot of these guys, they want to know, like, what does it cost? Do I get more feed? Do I get better feed? And here they did. So we've got a whole bunch of other examples on our website. We work all over the country. We do a lot here in Montana, obviously, California. Got folks in Illinois who do corn and soy, and then we do citrus in Florida, more corn in Nebraska. So we're all over the place. Yeah, thank you for having us.
You may need to say this again. So just for those of you in the room, if you would like to contribute to the conversation, you just activate your microphone by pushing down. So this is Julia, and I want to invite anyone to weigh in with a question. I think online, the best thing would be to raise your hand or put it in the chat, and Paige has her participant window open, and so she's watching that. I'll just use moderator's privilege to start with a question. So we're economic geographers. I'm really curious about the supply chain for these different products. And were you going to ask the same thing? Yeah. So where, who's manufacturing this worm tea? What's the issue with compost availability? Is there a meaningful, like when we think about sort of a holistic approach to business, is there a meaningful difference between the lab-made biologicals and the worm tea? I'm just curious, and who's poised to really, should this take off? Who's poised to kind of be in control of that industry, of the inputs, and just what can we learn about it?
That's a great question. So the products we work with out of Corvallis, they make it there. But then the folks we work with in Illinois, they make their own compost extract. So they take good compost, and then we help make it more. The hard part of compost extracts are making them fungal, and then extracting good fungi and also being consistent, combined with when you make your own, it goes bad fast. You got to get out there in like 12, 24 hours, depending. So the beauty of what they did at Earthford is he somehow has got a shelf-stable compost extract. So the folks in an almond orchard in California, they can order a semi-truckload, all they need for their own season, and then they inject it through their irrigation systems real easily. In Illinois, they're doing even more acreage, and so it'd be really expensive shipping all that water back and forth. So they've got their own compost extract machine set up. We help make that compost more fungal with Revive, and then they make a whole bunch really fast in the spring and put it in furrow for them and all their neighbors that they service there. So there's a whole variety. We don't really care which way folks do it. The same with like the worm teas. There's lots of people out there that make vermicasting and they extract the worm teas. So that's just another kind of version of Provide. And that's got the same problem if it goes bad. You have to have somebody locally who knows how to do it. But yeah, folks want to make their own, that'd be great. And if it blows up, Earthforce not going to be able to provide all this for the whole country. There'll need to be a whole bunch of companies doing it and folks doing it on their own. And there's a variety of tools like the fish. There's fish all over. There's tons of fish companies. We work with one out of Illinois. They're neat because they harvest invasive carp out of Mississippi. So taking a pest, a problem, and turning it into a solution. So we like working with them. But there's fish byproduct all over.
Huh.
Does that answer part of it? Yeah. What do you want to say?
So I got a question. When you say make something fungal or all these products and from compost, what fungi are you shooting for? And in other words, do you have specific species that are better than others? And what are the targets?
The goal is diversity of fungi. So we're not growing specific.
It's not mycorrhizal. It's the free floating type of fungi. And there's thousands of species. We want to have every player out there. We're not trying to like phytoderm. We're not trying to have one specific one. We have that's in there, but we just don't know who are the most significant players. We haven't done enough research yet. And Jed was telling us.
Oh, go ahead.
Jed was saying that there's some that are very small in numbers that have a huge role, that they're discovering that if they're not there, the synergy's not there. So we stay away from the lab. Produce stuff right now.
And then what tests do you use to find out if your compost worked or didn't work? What are you actually testing?
To see if it became more fungal.
Yeah.
So that's the earth fort direct microscopy. They take a sample and they actually count it, like humans in there counting bacteria and fungi. So you can send samples into the lab and they'll do it. For example, the folks in Illinois, they got good enough with a microscope and they sent enough samples. Now they're calibrated. When they make their compost, they make their extract, they can pull a little bit and say, this has a reasonable amount of fungi in it, or there's no fungi in it. So they are able to test their own stuff because they're in a tight window. They got to get it out immediately. They can't send them all in to get tested.
Thank you.
Questions online? No more none online.
Does anyone online want to ask a question?
Yeah, Tim McDermott here. I apologize for getting in late on your presentation. I raced home, tried to be on time, but I just couldn't quite get here. I'm about 15 minutes late. So keep that in mind when I ask the following question. You may have already explained it. How do you guys measure bacteria to fungi ratios?
We're doing it with direct microscopy. The lab we work with out of Oregon, Vern talked about at the beginning, we send in soil samples, just like you take a regular chemistry soil sample, but you just send it into the lab, 2-day shipping, get it there quick. And then they're pulling samples from that and doing counts under a microscope. There's, it was Dr. Elaine Ingham helped pioneer that method. And there's a whole bunch of studies out of Corvallis, I think, that I tell you exactly how they do it. I honestly don't know exactly how they do it. So.
Do you know the units they use? Like
they're
biomass or
like
Parts per million.
I'll find one of these. It's like it's whatever UG per G is. Yeah.
micrograms per gram?
Yeah.
Okay.
Mass, measure mass.
So they do some sort of count, and then they have equations that they use to get it to this.
Okay. Thank you.
And then the flagellates and the ciliates are doing like number per gram. Good question though.
I'm gonna read this into the microphone.
There's a question online from Amelia who asks, do the samples have to have specific conditions while arriving to the lab?
Another good question. You how much time do you have after taking a sample, and what conditions? Ideally, the ground can't be frozen. You don't want it to be like particularly flooded or oversaturated. So just a normal production day. We like to sample in this growing season so we know what's going on while you're trying to grow your crops. And then if you take it to the post office and do 2-day shipping, that's generally plenty time. You don't want to leave it in your truck on the dashboard over the weekend and 100 degrees. Biology is constantly changing. And then just for these tests, it's just a snapshot. It's a moving target. It's constantly changing. Biology is so dynamic. We never want to take just one test and go too crazy with it. So, take a couple tests, do some things, compare that with your actual production, forage quality, all the other metrics that we were looking at. So this is just one piece of the puzzle.
Oh, hello. As Julia mentioned, we're social scientists, so you guys have given us a pretty convincing presentation today, and I'm wondering what are the barriers to producers adopting regenerative practices?
That's a question. It's a great question we think about every day. The people are used to doing things a certain way. A lot of people we're talking to have been doing it the same way since longer than I've been alive, and it's hard to switch. There's also some just technical things. Like there's a whole system set up of people getting there. There's all these co-ops and utility companies, and it's very easy for them to do it the way they're used to. That you can just make a phone call and someone will come put it out on their field. So in Paradise Valley, folks are used to making a phone call and then the truck will come and just do it for them. For ours, you got to either find somebody to apply it for you with a boom sprayer. We do liquid, but it's getting easier. Now a lot of folks have switched over to putting injector systems into their pivots, which saves them a ton of money, and they can just apply exactly what they need. So our products will all go through injectors. So things are changing. Yeah, just people's mindsets, and then little logistical things. And then there's also just not a lot of, it's grown a lot since we've started doing it. I'd say we've been working on this for 4 years. And even in the last 4 years, the amount of folks talking about biologicals and soil biology, like the industry is growing, and we're just hopeful that people will find the good products out there and then implement them well. So when people do end up eventually trying it from us or from somebody else, they give it a good shot and they don't buy something that's snake oil or implement it wrong and then go right back to what they're doing.
Any like stigma associated with it? Like, oh, that's for hippies or something.
We have one of our first customers in Paradise Valley. He always calls it hippie fertilizer. So he's like making fun of it, but he's the one who his production's been going up every year from it. So he's like, yeah, it's funny, but he likes it. So
Nice. Okay, I'm going to sneak one last question in here before we close. So this is from Dr. Adam Sigler, who I don't know if you know Adam. He's an extension. He's a water specialist. He asks, for an experiment like the pivot, have you considered splitting the pivot into something like 8 pie pieces with alternating treatment and control? That way you could better address the possibility there are native soil differences across that space. And he says any other randomized approach for applying the treatment.
We talked about that a lot when we were setting this one up because Jed helped us and he also suggested that. But we're already asking these producers to do a lot of extra things. And so we wanted to split it in half so we could count the bales. But yes, in an ideal world, we would set up more replicates and we would do all of our, side-by-side trials more rigorously. So hopefully if we get some more funding, and we're applying for more grants, we're working with Jed on this, and anybody here who's interested to help us, we'd love to set up more rigorous trials like that. It's just more of a implementation thing.
Yeah, I was thinking maybe there's someplace at the post farm that is ready for this. I have no idea. It's not my space. But well, I need to close to respect everyone's time. It's 1 p.m. So thank you so much for a fascinating presentation. We hope to see you again. And thanks everyone for being here. We'll see you next week. Kathy Whitlock is going to share the results of the Greater Yellowstone Climate Assessment. So be sure not to miss that. Thanks everyone. We'll see you next time.
Thank you guys.
Good job. Thank you.
Thanks for coming.